My name is Diederik Morales. I'm a marine environmental biology and policy major at Monmouth University. And here's my poster describing how coastal lakes of Monmouth County have problems. So cyclically for the last many years there have been problems with harmful algal blooms in these lakes. And to be able to understand them and characterize them a little bit better, we need to understand the taxa that are causing these blooms. And typically the way you would do that is looking at them on a microscope, but we are using more technologies to try to do that better with environmental DNA, or eDNA. We collect samples and we identify every single taxa that's present in the water, and we simplify that into a community composition instead of looking at the individual taxa, and we get a cluster like this using a clustering technique called NMDS. And basically what it shows is similar community compositions cluster or group together in the same area. And we assign a color and a numeric value to those clusters so we can show them on a map to show the time and spatial scales of the community composition. So on a map right here you can see the differences in the fall between October and November. There was very few community changes because they're not as active. There's not a lot of reproduction happening, so they're staying very similar. But then in the summer of the next year, there's a big composition change, and that is likely because certain bacteria are growing more than others, so the composition changes. And some of these sites were blooming. And then a month later in August, the composition again changes, and we see that the blooms that are happening on the east side of Deal Lake and in Sunset have very different compositions, and they look different at the time, and that just shows that different taxa create different blooms. And knowing that now, we can kind of understand it a little bit better. And even though these were a relatively few samples, only 27 samples across 8 stations, this was a very important step because we created a pipeline where we can feed more data down the line. I just got back 47 samples from the lab, and we're analyzing the data to be able to include it in this. And within the next couple of weeks, we're about to send 180 more samples from 2023, again, to understand better all these changes in community composition. And hopefully in the future, we can even maybe understand how those changes relate to changes in the environment, or like rainfall data, changes in salinity, changes in certain things about the water. And hopefully we can use that data someday to forecast or predict harmful algal blooms.